structure of DNA, there are two different kinds of nucleic acid. First one is DNA, second one is RNA. This DNA stands for deoxyribonucleic acid. Then why the term deoxy here? Because when you see ribose sugar and deoxyribose sugar are the 2' carbon atom, one oxygen is absent. Due to absence of oxygen at the position 2, it is known as deoxyribose sugar, and the name is given as deoxyribonucleic acid. Its double helix structure was proposed by J.D. Watson and F.S.C. Crick in 1953, basing on X-ray diffraction studies of M.H.F. Wilkins. According to this structure, DNA consists of two polynucleotide chains which are anti-parallel to each other. Now, if you see these two strands, one strand runs in 5' to 3' direction, the other strand runs opposite to it. It means if you see the sugar phosphate backbone, one strand runs in such a manner that this one it will be the phosphate position at 3 carbon atom, and a nitrogenous base are attached to the carbon one of sugar. Now in the other strand, when you will see this one, under condition, the position of the strand will be in this manner. So the two nitrogenous base of adjacent strands will come closer to each other and they will form hydrogen bonds between them. Now the diameter of the DNA helix is 20 angstrom. And length of each helical turn means, suppose this one is the root and I am coiling one iron rod in this manner and another one rod at 20 cm distance in this manner. The two strands, they will look from the opposite side that they touch each other, but actually they do not touch each other, so they are anti-parallel in nature. And one turn means this one up to 180 degree and another 180 degree. Now this is the total turn. So length between this point and this point is known as one turn and its length is 34 angstrom. There are about 10 base pairs. 10 base pairs between each helical turn. So the distance between two subsequent base pairs will be 34 divided by 10, which is equals to 3.4 angstrom or 0.34 nanometer. Now another one property of this DNA double helix is the strands are complementary. That means if we know the sequence of one strand, we can calculate the sequence of other strand. Now see, suppose this is the first strand, then what will be the second strand? The second strand will be this one A, and A always pairs with which one? T. So this one will be T. Then this one T, it means it will be A. This one G means C. C, it will pair with G. Then C, it will pair with G, and another one A. So the other strands sequence will be which one? In 5' to 3' direction it will be A, G, G, C, A, T. According to Chargaff's rule, total number of purines is equal to total number of pyrimidines. Then what is purine? Purine, it is a 9-membered ring compound containing nitrogen at position 1379 and other positions are occupied by carbon. There are two different kinds of purine. One is adenine and another is guanine. They are purines. Similarly, pyrimidines. What are they? They are 6-membered ring compounds containing carbon and containing nitrogen at 1 and 3 position. And there are 3 different kinds of pyrimidines. One is cytosine, another one is thiamine, and another one is uracil. From these 5 nitrogenous bases, adenine, guanine, cytosine, and thiamine, they are present in DNA. And adenine always pairs with thiamine by help of double bond. Guanine always pairs with cytosine by help of triple bond. So according to Chargaff's rule, total number of purines is equal to total number of pyrimidines. It means A plus G is equal to C plus T, which is equal to 50%. If this number is not same, that means A plus G is not equal to C plus T, then the DNA must be linear DNA. Otherwise, the DNA which is present, it will be double stranded DNA and it will be linear or circular. If it is A plus is not equals to C plus T, then it is single stranded DNA, which is usually seen in case of viruses, not in the other organisms. So, what are the features of this DNA double helix? First one, the DNA double helix consists of two polynucleotide chains which are coiled around each other in right-handed manner around one imaginary axis. Then, diameter of the helix is 20 angstrom. Third one, length of the helical return is 34 angstrom. Fourth one, major groups. This one is major group and this one is minor group. Major group and minor groups are alternate with each other. And strands are anti-parallel. It means one strand runs in 5' to 3' direction. It means the sugar's 5 carbon is towards the opposite, and other strand in opposite direction. It means the sugar's 3' region will be towards opposite. So the two strands are anti-parallel, and the sugar is attached to nitrogenous base at carbon 1. And if you will see that DNA strands are complementary, it means adenine always pairs with thymine by double bond, guanine always pairs with cytosine by triple bond. And we can tell that according to Charcot's rule, total number of purines is equal to total number of pyrimidines. Length of DNA helix is 6800 angstrom. Then calculate total number of base pair. Second one, if it contains 20% adenine, calculate cytosine. Then third one, calculate total number of phosphodiester bond. These are the usual questions which comes in PMD. Then first question is answer. Total number of base pair present in it. We know that length of each helical return is 34 angstrom. Then the total length in the question is given as 6800 angstrom. So total number of turns will be 6800 divided by 34, which is equals to 200 turns. Each turn contains 10 base pairs, so total number of base pairs will be 200 into 10, that is 2000 base pairs. So answer is first one. Then the second question, if it contains 20% adenine, calculate cytosine present in it. Now there are 2000 base pairs. 2000 base pair means it is equal to 4000 base. And according to the question, there is 20% adenine. And our Charcot's rule says that adenine plus guanine is equal to 50%. It means adenine 20%, guanine 30%. So guanine forms triple bond with the cytosine. It means cytosine will be 30%. So total number of base is 2000 and its 30% is required. Its 30% is this one. It is 1200. So total number of cytosine will be 1200. Now the third question, calculate total number of phosphodiester bond. How many base pairs are present? That is, answer is 2000 base pairs. So in the first chain it will be 2000. In the second chain it will be also 2000. Now what is phosphodiester bond? Nucleotides are joined with each other by phosphodiester bond. It means suppose this one nucleotide, it is another one nucleotide, and in between this one phosphodiester bond exists. If it is the third nucleotide, then here another one phosphodiester bond exists. So between three nucleotides in a chain, there are two phosphodiester bonds. And according to the question, there is 2000 base pairs. It means the first strand contains 2000 nucleotide and the second strand also 2000. Now the phosphodiester bond in this strand will be 2000 minus 1, which is equals to 199. And this one, it is also 2000 minus 1. So the total number of phosphodiester bond will be 3998, which is also in this question. A DNA contains 242 adenine, 242 thymine, 143 cytosine and 142 guanine. Then this DNA is first option human DNA, second option bacterial DNA, third option viral DNA, and fourth option non-viral. Now this answer is, it is a viral DNA. Why? According to the bonding pattern or complementarity, adenine always pairs with thymine. It means there are 242 adenine and 242 thymine. So this rule satisfies the double stranded helical structure. But guanine must pair with cytosine. But in the question there is 143 cytosine but 142 guanine. It means cytosine does not pair with guanine. It means the DNA is not a double stranded DNA, so it is a single stranded DNA. Single stranded DNA exists in case of virus, so its answer is a viral DNA. Now you may think that here adenine is equal to thymine. Guanine is not equal to cytosine, then why it will be double stranded? Because according to Chadov's rule, total number of purine must be total number of pyrimidines in a double stranded structure. Now here adenine is 242, but guanine is 142. Similarly, cytosine is how much? This one it is 143, and thymine it is 242. So these two numbers or sum of the two numbers are not equal. It means it is not a double stranded DNA, which is a single stranded DNA. And single stranded DNA is the characteristic feature of virus only. So answer is this C.